We were then talking last time about more detail into T-cell based immunity, and now we turn to more detail on B-cell based immunity. And we had talked then about some of the details of this signal transduction pathways that are initiated in T cells when the T cell receptor encounters its cognate antigen. And so we're going to begin now with more details of B cell signal transduction when a mature naive B cell with IgM all over its surface encounters its cognate antigen. And so I mentioned to you that all growth factor receptors in cells need to, it's not sufficient for one growth factor receptor to bind a growth factor. That by itself cannot initiate a signal. Instead, we need to have at least two growth factor receptors, which are mobile in the plane of the membrane, encounter growth factor receptors, and then the binding of receptors will lead to the formation of dimers. The growth factor receptors dimerize. Okay? An equivalent thing happens with T cell receptors, basically. And we see the same thing now here with the immunoglobulin that's expressed on the surface of this mature naive B cell. Now, I guess we need to back up a step here. We said that the T cell, we said that there's a semantic thing here with T cells and their T cell receptor. Sometimes when we do speak of the T cell receptor, we mean the alpha, beta, or gamma, delta pair. But depending on the context, we actually mean the functional receptor that can receive signals. So that's the T cell receptor alpha beta chains plus the so-called CD3 proteins and the interior zeta chain. In a similar way, for the IgM here, which is basically the antigen is the growth factor binding to this receptor here. And now, because the receptor itself has just like a few amino acids inside the cell, it can't really by itself transduce and recruit signal transduction molecules. It, like the T cell receptor, the CD3 complex, we have, as we mentioned earlier in the semester, these immunoglobulin alpha and beta chains here that are associated with PTL, and they have longer transmembrane, they have cytosolic parts inside here that are long enough to recruit other partner proteins and so on. So the actual B cell receptor, as opposed to just this IgM molecule, a B cell receptor is an IgM plus the Ig alpha and Ig beta companion proteins. Okay. Now, if we have pretty closely spaced antigen here, the closely spaced antigen will cause the immunoglobulin M's, or just let's call them the B cell receptors, to then be practically oligomerized here. They're basically they're so close to each other that that is sufficient to initiate the signal transduction events, the recruitment of Src family tyrosine kinases to initiate events here. Okay. And here's then the same we'll call it horrifying picture that we kind of showed when we went into the details of T cell receptor signaling. Not so horrifying because as I told you, for T cell receptors, I'm not going to hold you to being able to reproduce all these pathways on an essay question or something like that. There are a few key things you should know, but I told you, I talked about them. case of the T cell receptor, and again, we're going to turn on possible like C gamma, PI3 kinase, gamma kinase pathways as we did to the T cell receptors. Remember, the phospholipase C gamma pathway mobilized internal calcium. It opens calcium channels in the membrane of the endoplasmic reticulum. Remember that calcium is released and does a number of things. In the case of T cells, you may remember it activates the phosphatase phase. NFAT is not in B cells, so the calcium does turn on other pathways, including protein kinase C, but we're not going to worry about all that. You may remember in the case of the T cell receptor, kind of an apex kinase. It was called ZAP70 in the case of T cells. So Src family kinases start phosphorylating things, and we recruit then the ZAP70 by its SH2 domains to phosphorylate tyrosines. In B cells, the equivalent kinase is a Src family kinase called SYK, which stands for Y as an abbreviation for the amino acid tyrosine. So this is spleen tyrosine kinase. Okay. And it sits here, it has SH2 domains. Src family kinases become activated and tyrosine phosphorylate the Ig alpha and beta chains. The SH2 domains of SYK bind those phosphotyrosines, and then they in turn get phosphorylated by Src family kinases, and thereby activated, and then the whole parallel cascade of pathways is initiated. Now, there's a very important, about the time we first discovered, getting on for decades, that there were protein kinases, enzymes that phosphorylate target proteins. In all cases, those so-called protein kinases, I told you before, phosphorylate serine or 3-adene hydroxyl groups, the side chains there. And then in the 1980s, we discovered that there's actually a tyrosine kinase activity that cells have. Very rare. It comes into play only in growth signaling and developmental differentiation events. Src was the prototype, the first one we discovered. And then shortly thereafter, we discovered that there was a special class of protein phosphatases. These are protein tyrosine phosphatases, phosphotyrosine phosphatases. And one of the famous ones found in lymphocytes is the CD4, the CD45 phosphatase here. And the CD45 phosphatase comes into play in terms of activating the latent Src family tyrosine kinases that are going to come into play here. In particular, we're talking about FIN. Blick, FIN, and LIN are all protein tyrosine kinases that closely resemble the Src itself. Few amino acid differences here and there, sometimes different ways of being activated a little bit. But so they are, so in particular FIN I think is an important. But also Blick, a B cell lymphocyte kinase here, they become activated and they start phosphorylating the Ig alpha beta tails. They're going to recruit the SYK tyrosine kinase. Okay. Now down here, I'm going to show you a crude representation of Src and all other Src family tyrosine kinases. They have an amino terminal end, which is post-translationally modified to put on a 12-carbon fatty acid called myristic acid, saturated fatty acid. And so the amino terminal end is myristylated, and consequently, that myristate inserts into the phospholipid bilayer of the plasma membrane. So all Src family kinases are, if this is the membrane, they're all just right on the inner surface of the membrane, held there by that myristate and the fluid minor injection, and they're just gliding all over this surface, the interior surface of the plasma membrane, and they get recruited So this is the immunoterminal end. There is a conserved sequence that began turning up in lots of other interesting developmental growth-related proteins, cytoskeletal proteins, called an SH3, Src homology 3 domain here, which binds certain proline residues on partner proteins. And then we continue into a so-called SH2 domain. Of course, we've been talking about SH2 domains on sideways the last few lectures. SH2 domains, you remember, bind phosphotyrosine, phosphorylated tyrosine residues here. And down near the carboxy-terminal end of Src, and Src itself is tyrosine 527. This gets phosphorylated by another tyrosine kinase whose special job in life is only to phosphorylate this, and by phosphorylating this, we inhibit Src. And Src is inhibited because there's an internal interaction between the Src SH2 domain and this tyrosine here. It's like Src sort of bends over backwards and bites its own tail, and that puts a strain on the catalytic domain here. So when Src is in this configuration, it is the kinase domain, TK for tyrosine kinase, is enzymatically inactive. And if we remove the phosphate here, then this pops off. We relieve this, the whole Src domain kinase straightens out, and we relieve the strain on the catalytic activity. So here's the Src, when the SH2 domain has released its grip on the active site here, and now it can then phosphorylate target protein. So it's a substrate protein. Active Src will now phosphorylate a tyrosine, or one or more tyrosines on the substrate, which when put on this, and when the tyrosines are phosphorylated, that typically then activates an enzymatic activity associated with the target protein. Now I'm going to show you more detail on this. By the way, and what we said up here is that the CD45 phosphotyrosine phosphatase activates Src. Well, here's where the CD45 is an integral membrane protein you saw. And you see, well, it turns out that this SH2 domains, many proteins have SH2 domains, but those SH2, while it's true that all SH2 domains bind phosphorylated tyrosine residues, the amino acid context on either side can make a difference, a big difference. So SH2 domains prefer certain phosphotyrosines on certain proteins because in those certain proteins we have the amino acids adjacent to it. Well, this is a non-optimal. We do not have the preferred amino acid context in this thing here. So this doesn't taste particularly good. It'll bite it, and after a few minutes it'll spit it out, but it's right in front of its face, so it bites it again. So it's constantly biting it, releasing it, biting it, releasing it. In those moments where it's released it, CD45 will come in there, cleave off that phosphate, and now there's nothing left for the SH2 domain to bind to. And often it will get recruited, once we've activated Src, often Src will get recruited to a target because it'll have itself has been previously tyrosine phosphorylated by something else. The SH2 domain will bind that, and then Src will continue putting another phosphotyrosine. I'll give you more detail than you need to know, as always. So here, since my own research back in the day was very much focused on Src and Src kinases, I'm going to make you learn a little bit about Src. So here's a prettier picture for Src family kinases. So here's Src, sort of bent over backwards, biting its own tail here with this SH2 domain, which puts a strain on the catalytic domain. It's got two lobes and then a cleft in the middle. The cleft here is all a single polypeptide chain here. In here, the bottom of the cleft is the active site. Like almost all protein kinases, the tyrosine or serine kinases, there's often something called an A loop, an activation loop here, which blocks the active site. And there's a case of the kinases. This amino acid has a tyrosine in it, okay? And there's some positive charged lysines down here. When we phosphorylate, Src is beginning to activate. Src is often found as dimer pairs, and one Src will phosphorylate another partner on the tyrosine here. That introduces a negative charge on the A loop, which makes it fall away, and this phosphate is then kind of pinned by electrostatic interactions with these positive charges down here. Now we have free access to the active site. So here's the amino terminal N with its myristic acid. Here's the SH3 domain, come down to the SH2 domain, and then the catalytic domain, and then the carboxy terminal N with the inhibitory side of phosphorylation. So Src has an inhibitory side of tyrosine phosphorylation in the carboxy terminus, and an activating side of tyrosine phosphorylation in the middle of the catalytic domain on the matrix. And so here's CD45 is going to grab that phosphate off, gets an opportunity that will make Src family. I didn't say here. The SH3 domain prefers certain proline residues. There is such a proline residue in this connecting loop. So we have an internal SH3 proline interaction and an internal SH2 phosphotyrosine reaction, locking Src into an inactive proline. We release this string, that often then leads to releasing of this string here. And here's some of the protein that comes along. It's got the preferred polyproline site for SH2, and or it also has maybe a phosphotyrosine already. And now Src, now these internal interactions are completely broken. The catalytic domain is in a relaxed state. And then one Src can phosphorylate the other, partially active Src, and phosphorylate that activating tyrosine there. And now we have fully active Src able to phosphorylate either this protein or some other major tyrosine phosphatase target. It's a great pleasure over the course of my research career to see all the stuff worked out in molecular detail like this. It's fun doing the scientific, even if you're doing all the experiments yourself. Okay. So, to continue on talking about activation of B cells. So we began by saying a mature naive B cell, if it happens to encounter maybe a bacterial surface where we have closely spaced arrays of the containing antigen of that B cell, that will cause clustering of the B cell receptors, and that is then sufficient for the first step of activation. We're still going to have to get the T helper cell cytokines there. But now, it turns out that the B cells can be activated. You don't always have to have one way to activate them, is to have tightly clustered antigen. If we have widely spaced antigen, we still can get activated. And in particular, it turns out, well, towards the last half of the class, we're going to get started talking about complement and all its complicated glory. And I mentioned to you in the beginning of the course that complement is a set of proteins in the blood which are inactive, and then they can become activated by a protease cleavage of complement number 1, and activated complement 1 will cleave and activate complement number 2, etc., etc., etc. It's an amplifying cascade. In the end, we produce pieces of complement number 3. In this case, we just speak of C3, but there are also further cleaved subtypes called C3A, B, C, D, and so forth. So here's this piece of CD3 complement that became covalently bound to some macromolecule on the surface of this, let's say, bacterial cell. Now here we don't have closely spaced antigen, but we have complement pretty close by to the B cell receptor. Okay? And then we have a complement receptor here, complement receptor 2, CR2, and this serves as a kind of a co-stimulator. It's a little bit like, in terms of the T cell way of thinking, we had like a CD28 getting a B7 signal here. This is kind of analogous to the second signal we would get from CD28 in T cells. It's initiated by the complement receptor, which has two partner proteins. Okay, so this whole thing here, these three proteins, that's called the B cell co-receptor. B cell co-receptor is a complement receptor with a pair of partner proteins. And this gets, if this is close to the antigen, then CD19 is going to be finding itself close to a Src family kinase called LIN, lymphocyte kinase, and LIN will phosphorylate CD19, which will then recruit via the SH2 domain PI3 kinase, which will turn on the PI3 kinase pathway. We also turn on some other pathways, really not shown here, and we're not going to worry about that. Your book says, at least last time I read your book, that the function of CD81 is not known. So, again, I see last year I said in the review, not on the test. I maybe will just want you to remember that there is a complement receptor that functions as a B cell co-receptor. Okay. Of course, when the B cell becomes activated, as we've said a million times now, the antigen activation, coupled with the T cell, helper cell cytokines, especially IL-4, importantly, this B cell, which is essentially a nucleus with a little rim of cytoplasm around it, becomes a big cell with a bigger nucleus and a whole lot more cytoplasm, and the cytoplasm is filled up with rough endoplasmic reticulum. Why is the cytoplasm of this plasma cell filled with rough endoplasmic reticulum?
It's a giant antibody producing factory, so it's going to be
That's exactly correct. It's a giant antibody producing factory. And what does that got to do with the endoplasmic reticulum?
The endoplasmic reticulum is what to actually synthesize the antibodies.
Thank you. She said that whenever we are by and large, when any cell is going to secrete proteins, it makes it on ribosomes which are bound to the surface of the endoplasmic reticulum on little channels that go through the ER membrane into the lumen of the ER. And we read the messenger RNA off of those ER-bound ribosomes, and as we read the mRNA into the protein is co-translationally translocated into the lumen of the ER, and then it goes by vesicle trafficking through the Golgi compartment, and then it fuses with the plasma membrane and releases that protein to the outside world. So you need to have endoplasmic reticulum to synthesize secreted proteins. And this guy is synthesizing industrial scale production of antibodies to the outside world. RER, site of synthesis of secreted proteins. Okay. Now, there are some situations where if we get certain ancient infections, whether they're plaguing us vertebrate animals, the Trinity, we have streamlined the process of B cell activation. Okay? So for certain common infections where you want to mobilize the troops, post haste, you're going to, what has been involved is an activation event that does not require T helper cell cytokines, T helper 2 cytokines. These events and the antigens that initiate them, this is called thymus independent. These are called thymus independent antigens. These antigens are so found on certain bacteria, negative bacteria, for example, very often, which are just nasty beasts when they get into us, strains of them, that over time we have evolved a sort of a shortcut pathway that doesn't depend upon mobilizing CD4 T cells. And so there are two kinds of thymus independent antigens. Thymus independent 1 and then we'll talk about thymus independent 2 type antigens. Okay, the TI1 antigens. So there's a class of antigens which can activate B cells without the help of T helper 2 cells. One is thymus independent. They form 2 categories, TI1 and TI2. The classic thymus independent 1 antigen is lipopolysaccharide. Okay? We find lipopolysaccharide in the outer membrane of gram-negative bacteria, E. coli, Haemophilus influenzae, Morellosis, Salmonella. Test. But LPS is a very potent. Are you resistant to LPS? Red alert. DEFCON 1. Okay. And so, and as we talked about before, what recognizes proteins like LPS is the famous toll-like receptors. Okay. In this case, it's toll-like receptor number 4. Okay, so here in yellow are a pair of TL4 proteins here. Okay. And they work in conjunction with a partner protein called CD14 and ND2. Okay. Here's the gram-negative bacteria. Here's the lipopolysaccharide. Okay, some of it involves fusing off the surface of bacteria. And there's a protein in the tissue in the blood, the LPS binding protein here. LPS binding protein, when it encounters free LPS, it grabs it and then delivers it to the TL4 complex here as shown. So here's the lipopolysaccharide, part of it being bound by CD14, part of it being bound by this MD2 component. Okay. And this will then initiate signaling events. We talked about the signaling events initiated by toll-like receptors in previous lecture. It involves, among other things, the NF-kappa B pathway. As soon as you see this on the test, there's our thing about what you're going to do later on this afternoon and this evening. Do not pay attention to what I'm talking about. So, if this were a B cell whose cognitive antigen is LPS molecule, the same LPS which is binding to and activating the B cell receptor is also binding and activating the toll-like receptor pathway. And now we can just go right through a quite quick pathway here, and boom, we get proliferation and differentiation of plasma cells independent of any IL-4 T helper cells. But in fact, once we're other B cells recognizing other antigens can also, with their own TL4s, also. So we can get, so lipopolysaccharide will activate many other B cells seeing other antigens. Okay. Now, you remember we said that we have B1 and B2. Yeah, no, never mind. This is the usual B2 B cells. Okay. It doesn't have to be lipopolysaccharide. It can be bacterial DNA that can also be bound by certain toll-like receptor pathways and initiate the same pathway. Okay, so so lipopolysaccharide and bacterial DNA are antigens that permit us to are so potent that we can bypass CD4 T cell help. So now that there are the TI2 antigens, and Now, this is really a little bit like what we began talking about this afternoon. TI2 antigens are typically composed of highly repetitive, closely spaced carbohydrate or protein epitopes at a very high density on the surface of the microorganism, such as pneumococcal polysaccharide or the flagellum protein in bacterial flagellum. Okay? So tightly spaced antigen will cluster a whole bunch of B cell receptors, and that's sufficient to activate the pathway and strongly enough that we don't require the IL-4 cytokine. Now, typically, however, in these cases, with these TI2 antigens, it's these little primitive B1 that are activated in this way. B2s, B cells, are the main B cells we've been talking about. I mentioned to you in a previous lecture that there is some number of these more primitive B1 cells, and they fire off without CD4 T cell help. These highly tested, high density antigens. Okay. So this is a little table here summarizing T cell dependent antigens versus TI1, TI2. The T cell dependent, which get CD4 T cell help, these cells can class switch at that same process. The cytokines released by T helper-2s are what you need to switch from making IgM, IgG, whatever. These guys are making, they don't class switch, they just secrete IgM. Okay? And these B cells, we can not only class switch and affinity select and so forth, we can high affinity class switch genes. Those cells, some of those cells can then become memory B cells for immunity afterwards. Not here, okay? No memory cells, no class switching. Now we come to 7 pages in your lecture notes, and in this chapter of the book, which is this stuff that we already talked about way back on the first exam, what happens, what are the details of what happens. And the second exam, we had this question, what are the details of what happens in the lymph node when the T cell, the B cell encounters their cognitive energy and so forth. This is all a repeat. So pages 11-23, this page, through 12-1 down. I'm not going to lecture on it now. Take the time. We went through it. It's not on the test. All false ways right down. These pages being not on the test. It makes it read better. 11-23 through 12-1. Test. Covered all this in the lecture then. Okay, now then, let's talk a little bit more. We've talked about what do antibodies do, and here's a little summary of some of the things that they do here. The resting B cell makes surface IgM, it makes MHC class II molecules, and it does not secrete antibody. This is intrinsic. It has inducible growth, somatic hypermutation, and IgM to IgG or isotype switch. The plasma cell is the finished product. It can no longer proliferate. It's a fully differentiated, non-proliferative cell. It's doing its job until it dies. Okay. It does not express surface immunoglobulin any longer. It does not express MHC class II molecules. It has a high rate of antibody secretion, of course. No longer capable of proliferating, no longer capable of any further somatic hypermutation, no longer capable of switching isotype. So now we're going to talk about some of the properties of some of the different immunoglobulin isoforms, IgG, IgA, and then there are subtypes of IgG and so forth. Well, immunoglobulin G, for one thing, we do find immunoglobulin G in the blood, but most of the immunoglobulin G is actually in the tissue fluids no longer in the blood capillary, the blood flow, but moves into the tissue space where we have this plasma form of blood bathing the cells here. And so here is the lumen of a blood capillary with soluble immunoglobulin G, and there's a protein called the Bramble receptor, shown here in green. The Bramble receptor has a domain that recognizes the Fc tail of an antibody. So the Bramble receptor is an Fc receptor. It binds the crystallizable fragment of crystallizable domain of G molecules, and it does what's called transcytosis. It does an endocytosis event to bring this complex into the cell in the form of an intracellular vesicle, and that intracellular vesicle travels across the cell to the other side of the cell, where it then fuses with the other side of this side of the room to do exocytosis. So we do endocytosis and then exocytosis, and in that way we deliver the immunoglobulin G from the blood into the tissue space where it can diffuse around. It has been here in this. If it's in the blood, it's passing through the liver and the spleen, and eventually it gets degraded and so forth. But here it's protected, so it's much more like very long-lived in this compartment. So you will remember from the test about multiple choice or true false that the Bramble receptor recognizes immunoglobulin G. There's another receptor that performs a similar function for immunoglobulin A that we're going to talk about in a moment, but not quite yet. So here's the distribution of immunoglobulin isotypes in the body. Color-coded here in flesh-colored pink, I guess you can call this. We have immunoglobulin G and monomeric IgA. Okay. So IgG and IgA are in the tissue space here. All of this here and so forth. They're also, let's see, well, I'm just going to start reading here. Immunoglobulin G and monomeric IgA are in the tissues here. Okay. Dimeric IgA, or IgA2, is secreted across internal epithelia. So the pleural space, that little fluid in which, that little fluidy cavity in which your lungs sit, the surface of that is loaded with IgA. Okay? On the lining of your gut and so forth is all loaded with IgA, and you're sort of embedded in the extracellular matrix kind of stuff. Okay. The mother will secrete her dimeric IgA in her milk for the baby, so the nursing baby shares in the mother's immunity. Whatever pathogens the mother encountered, she's transferring those antibodies to her baby. The baby yet can't mount its own immune responses effectively, and has not yet encountered many of these pathogens that the mother in her life has encountered. So the baby is getting immunized against the environment the mother grew up in by the IgA2 in the baby's milk. Okay. Protects the baby from skin infections and gut protocol. IgM and IgG monomer K are in the blood. Okay. So these guys and these guys are in the blood circulation, as well as in the tissues. And the baby then also gets maternal immunoglobulin G being then moved across the placenta. Okay. The baby's getting IgG while it's in utero and growing, and then when it's born, it gets IgA in the mother's milk. So this is now these are the cytokines that T helper 2 cells release or T helper 1 cells release. Remember T helper 2, IL-4 we've been saying is B cell growth factor, of course. So it induces B cell proliferation, but also inhibits the production of IgM, inhibits the production of IgG3, induces IgG1 expression, inhibits IgG2A, IL-5, augments the production of IgA, TGF-beta, these things, interferon gamma induces IgG3 production and IgG2A production. Having said all that, on the test. Now, I told you just a minute ago, when we were talking about the Bramble receptor, that moves IgG out of the blood. The receptor that moves IgA out of the blood and into the extracellular space here is the so-called poly-IG receptor. So here's a plasma cell secreting dimeric IgA in the lemma propria. And then this dimeric IgA is free to diffuse across the basement membrane here. And now this poly-IG receptor, it's not, it's really just dimeric, it's hardly poly, but anyway, the dimeric IgA, known as the poly-IG receptor, will do the same thing, the same transcytosis. It grabs it and endocytoses it in as a vesicle. This vesicle just goes right to the other side of the cell, where it then fuses with the apical plasma membrane, and then we do exocytosis event, releasing the dimeric IgA, which becomes part of that little mucopolysaccharide coat that lines the surface of the gut. The gut here. We said that another important function of antibodies is to neutralize bacterial toxins. Many bacteria and other pathogens secrete certain proteins which kill our own body cells. I guess they do that because when the cells die, they release all their macromolecules and sugars, and then which is just food for the bacteria, basically. And we always hope there'll be some B cells whose antibody recognizes that toxin is a foreign antigen and will bind it and neutralize it. Now, if the toxin has to have two components to it. One component binds to a receptor on the cell surface that it's about to be able to bind, and that brings it into the cell. And then another function it has is to bind to something and kill some enzyme inside of the cell, kill a ribosome or whatever. So it has a catalytic domain and a binding domain. We'd like the antibodies ideally to bind to the binding domain so that the toxin cannot bind to the surface of the cell, and then so therefore can't get into the cell. Those would be neutralizing antibodies. Other antibodies would bind somewhere. They might bind to the protein, but but uselessly they don't prevent binding and they don't prevent entry into the cell. So here's a whole table of all kinds of bacteria and toxins they produce. I'm not going to worry about that for the test. Here's an interesting little picture on the baby. So here's the level of fraction of adult level of serum antibodies inside the baby, starting at about, I guess about here, 6 months or so. And we see that the baby is starting to make its own IgM a little bit. Okay? It's got the passively transferred mother's IgG. And then here's birth. Just before birth and right afterwards, we have an exponential increase in the baby being able to make B cells with their own IgM. And with the lag, the baby's ability to make B cells that can do IgG. Okay? And finally, the baby's own IgA. Okay? This doesn't show the maternal IgA that's coming in by the milk. It'll be right up here, just after birth. There's a period here you can see, in about 3 months, where the baby's is losing the mother-derived immunoglobulin G, and it's not really quite come up to par here with its own synthesis of IgMs and IgGs. So there's a little overall low level of protection here at about 3 months. So good time for the mother to say, we're not going to go to the mall this weekend. The next door neighbor, you can't come kiss the baby yet. Here's a picture of it. Just illustrates what I was saying about. Here's the toxin, yellow and red. It has a yellow binding domain and then a red catalytic domain. So it binds to some kind of peptide that's evolved to bind to. It's brought in, and then in various ways it will cleave or release catalytic activity of the toxin inside the cell. And ideally, antibodies will block the binding, the yellow binding domain, so that it can't get into the cell to begin with. In addition to blocking toxins in this way, we can block viruses this way. Viruses are what we'll call viral spike proteins, which have evolved receptors on the surface of the cell, which will result in the virus getting in. Ideally, we have antibodies that bind the viral spike protein and bind just the right place on that viral spike protein so that it cannot bind its cellular receptor. That would be an antiviral neutralizing antibody, for example. It's very difficult for us to get neutralizing antibodies against the HIV virus, so we don't really have effective vaccines yet. The HIV virus is strictly a little devil, and we can't seem to it mutates away from attempts to make vaccines. Now, so what is the point, then, of having IgA antibodies or IgG antibodies or whatever? Well, the point is, in addition to the things we've been talking about here, neutralizing activities of antibodies, and what we didn't talk about, which is the fact that the antibodies coat the surface of the bacteria. Anybody remember what that term is for coating the surface of the bacteria?
Optimization.
Many different cell types respond to the presence of a particular isoform of an antibody by recognizing it's the tail of the antibody. Remember, when we do class switching, we keep the antigen binding part, and we just switch out the tail. And different cell types have transmembrane protein receptors for the tail of IgA or IgG and so forth. And then these Ig receptors initiate signal transduction pathways inside of cells. So, for example, the oxidization business here is triggered by a particular Fc receptor. Okay? And then these receptors, when they bind the tails and immunoglobulin Gs are often very good at oxidizing. As an Fc receptor, we call this an Fc gamma receptor. Gamma is the Greek letter for G. Okay? And the Fc receptor, Fc gamma receptor here, that macrophages express, when it binds the oxidizing antibody, that initiates a signal transduction pathway that then leads to the engulfment and phagocytosis of the bacteria. So here are, this is a complete list, but here are the more common important Fc receptors. They all have the Greek letter here. So if we're recognizing immunoglobulin G, we would speak of an Fc gamma receptor. And we have a set of Fc gamma receptors from numeral 1, from numeral 2A, from numeral 2B2, and so forth here. And they have associated either partner proteins or internal domains that initiate signal transduction events. And these can include sites for tyrosine phosphorylation. And, here are the particular, look here. So these were immunoglobulin. I'm recognizing IgG molecules. They're all Fc gammas. Here's an Fc epsilon. Epsilon is Greek for the letter E. So this receptor binds immunoglobulin E. And we talked about the fact that mast cells have got hundreds of thousands of copies of this Fc epsilon receptor, which will bind the tail of an IgE. And so whenever all the different, you're in certain parts of the world where you get all kinds of parasites and worms and so forth, the mast cells are, the way we fight that is to have the B cells class switch to making IgE. The B cells secrete the IgE. It is then bound by these Fc epsilon receptors on the mast cells. So if the IgE against antigen 1 is colored pink, later on you encounter another pathogen making an Fc with that pathogen, make that purple, and over the years the mast cells become coated with like a palette of colors of all different IgEs against all the different parasites it's encountered. So that if you later on encounter it, then that is going to find one of it is going to find an IgE bound to the surface of that mast cell, and the parasite will then cause the IgE via the Fc epsilon receptor will initiate this signal transduction pathway that causes the exocytosis of naxitotic stuff on the surface of the worm. Okay. Look at the relative affinities here. The binding. So this is some arbitrary units here. Pretty good binding of IgG here. Rather modest binding here. Actually, these guys sent an image. Look at here. 20,000 affinity here as compared to, the other way highest was 200. So FCEs, when 10 years ago, some was made and bound to some of the FCE receptors on the mast cell, it's still there, 5 years later or whatever, 10 years later, they find it so tight. So here I made a note. You will just know about the Fc epsilon receptor on mast cells for the test. Binds IgE. And then here's a little bit more elaboration on this. Here's all these seven shown here. Many more. These are all Fc epsilon receptors. And these are all the different IgEs, IgE against X, IgE against Y, IgE against Z. And this is what the resting mast cell looks like. All these granules here, we've got some nasty SHIT in them, but they make the worms unhappy, and so you don't see it, but here's what's on the surface. And then along comes some parasite that we've seen before, and we already have ready-made IgE against this little parasite component here, and kaboom, massive exocytosis of all these granules here. There's a picture here. Here's this little schistosome, nasty creature. Here are these eosinophils and mast cells all over the surface, and they're just raining down this nasty stuff onto the surface of the schistosome and making life miserable for it. I guess I'll go back here for a moment here to the preceding page 12. So, the function of mast cells in IgE is to fight parasites, we secrete in these granules here. We have histamines, proteases, various cytokines, chemokines, prostaglandins that cause inflammation, smooth muscle contraction. Mucosal epithelial cells start secreting mucus. Okay. Basically, all this stuff is designed to produce for your body just to mount huge mucousy fluid production and just wash the stuff out of your airway and your nostrils and so forth. And of course, the price of the modern world we live in is that you don't really encounter these things in childhood, and so the only thing these things could ever do for fun is to respond to the pollens coming down from Oklahoma and everywhere else north and west of us here, so we get seasonal allergies. And we take antihistamines to prevent the symptoms of the allergy, of course. Yes, there's one more very important and famous function for Fc receptors. We'll talk more in time about a cell called a natural killer cell. Natural killer cells come from the same lineage as the B cells and the T cells, but their main function is to fight virus infections early on, in the time that it takes for the adaptive immune system to come into play to fight the virus. The natural killer cells are, defending the bridge. They're really holding off the onslaught of the barbarians, and they do that effectively in various ways. But then once we have the adaptive immune system going and we're making antibodies, they continue to play an important role because they have evolved an ability. So here's a virus infected cell, and so we're making viral particles and releasing them from this cell. So the surface of this cell has partially formed viral particles and so forth, and antiviral antibodies will bind to those partially formed viral particles that are part of the plasma membrane of the cell, and so the antiviral antibodies binding the surface of a virus-infected cell. And there's something called antibody-dependent cell-mediated cytotoxicity, ADCC. And here's the deal: the natural killer cells, they have these Fc receptors, and they bind the tails of the antibody, and then the Fc receptor initiates a signal here that's going to cause the release of these granules here. This is very much like the way that killer T cells will kill a cell. They have the same kind of granules, apoptosis-inducing granules that will be then sent into the target cell and cause it to die by programmed cell death. Okay. Now we come to complement. Here's another big role antibodies do for us to help us fight infections. And we're going to take break now. When we come back, I'm going to tell you that there are 3 ways in which this blood protein complement cascade process can be initiated. 2 of the ways are independent of antibodies or the immune system. They are part of the innate immunity, basically. The lectin pathway and the alternative pathway. But even once as long as we're fighting with antibodies, we continue to activate complement, this time based on the antibodies that are being produced. So antibodies bind to the surface of a bacteria or affected cell or something like that, and induce the activation of the complement cascade. So let's take a break, and then we'll come.